Okay, so this video is going to be on the second half of Module 7 for biology. And before we jump into the new material, I wanted to do just a quick overview of protein synthesis used from DNA instructions that we talked about last time. So remember that we said that DNA is present in the nucleus, and in order for a particular protein to be made, the part of the DNA that gives instructions for that trait or characteristic unwinds so that you no longer have the double helix structure. So let's say that the bases that So let's say this is a strand of DNA in the nucleus. And then what we said happens is that a type of RNA that is called messenger RNA will take a picture of this DNA in the nucleus, and it takes that picture by lining up its appropriate bases. So C will join with G, G will join with C, C with G. A, remember that RNA does not have thymine, it has uracil. So the A will bind with uracil, C with G, T with A, T with A with uracil, and C with G. Okay, so this would be the strand of messenger RNA that will carry the message of the DNA from the nucleus into the ribosome. Okay, so this strand of RNA will now leave the nucleus and will go to the ribosome. Okay, then when it gets to the ribosome, it will meet up with, it will combine with transfer RNA, and it will do that by connecting the appropriate bases. So the messenger RNA combines with the bases on transfer RNA. So G with C, C with G, G with C, U with A, G with C, A with U, A with U, U with G, and G with C. Okay, so where you would have, where U with A, there's no thymine, so this would be the transfer RNA that gets made from this strand of messenger RNA. The other thing we said was that on both messenger RNA and transfer RNA, these bases tend to group themselves into groups of 3. And each of those groups of 3 on messenger RNA, each group of 3 is called a codon. And on transfer RNA, each group of 3 is called an anticodon. Okay, so we started with these instructions from DNA, and now we have the instructions in the form of transfer RNA to make the protein dictated by this strand of DNA. And then the last thing we said was that each of these anticodons carries with it a particular amino acid. So that amino acid might be leucine, one might be tyrosine. There's 20 different amino acids. Maybe this is another leucine. So every three groupings corresponds to a different amino acid. So since these are all 3 different, then they would all need to be different amino acids, so we can make this one alanine. So as these bases are lining up with each other in groups of 3 on the transfer RNA, we are also lining up amino acids. So depending on how long this strand of transfer RNA is, that determines how many amino acids get lined up and in what order they get lined up, and that's what protein is. So the instructions that came from DNA to build a particular type of protein is now accomplished by the lining up of these anticodons and the amino acids that they carry with them. Okay, so that's just a quick review of the process of protein synthesis that we talked about last week. So be sure that you can be sure that if you're given a strand of DNA, be sure that you could say what the messenger RNA and transfer RNA would look like for that particular strand. Okay. The second half of Module 7 then deals with 2 processes that happen in the cell, and those processes are called mitosis and meiosis. Okay, so what is the difference between the two? Mitosis is asexual reproduction of cells. And meiosis is sexual reproduction. So sexual reproduction by organisms that result in offspring. Now, previously in this course, we have talked about asexual reproduction as the way that things like bacteria or certain organisms like sponges or some fungi reproduce. However, the thing that I want you to realize is that in organisms that reproduce by sexual reproduction, all of the cells in those organisms must be able to reproduce asexually. And the reason for that is it is through asexual reproduction that organisms grow. Okay, so when you all were born, most of you probably weighed somewhere between 6 and 8 pounds. And how did you get to be 100 pounds? Well, the answer to that is that your cells had to reproduce asexually. So you start with 1 cell, that 1 cell becomes 2, 2 becomes 4, 4 becomes 8, etc., etc. So growth takes place by asexual reproduction of cells. And then in addition to that, our bodies also replace dead cells by asexual reproduction. So your red blood cells live only about 3 to 4 months. So your body is having to constantly be replacing red blood cells that have died, and that they do that by mitosis. Or every time you touch something, you have dead skin cells that are rubbing off or getting sloughing off of your hands onto whatever it is that you touch. So our skin cells are constantly dying, and those cells need to be replaced, and they are replaced by asexual reproduction of skin cells. So growth of cells and or growth of organisms and replacement of dying cells happens by mitosis in organisms that reproduce, even in organisms that reproduce offspring by sexual reproduction. The cells still need to be able to do asexual reproduction. And then sexual reproduction is the process by which organisms, by which the whole organism gets reproduced. So for meiosis, you need to have a male and a female. In a male, the cell is called a sperm. In a female, is called an egg. And the offspring have characteristics of each of the parents. So we might have eye color from the female and hair color from the male, but there's the traits are a mixture. The offspring will be a mixture of traits from each of the parents. Unlike mitosis, where the cells that are reproduced in mitosis are exact copies of the cells from which they came. Okay, so what we're going to do then is we're going to look at each of these processes, and before we do that, the first thing that we need to talk about is a chromosome. So you might wonder, I think I've mentioned that if we were to take a strand of DNA from a cell, and we will do this as a lab, but if you were to take a strand of DNA from a cell and stretch it out, that one strand of DNA could be 6 to 8 feet long, and yet it is crammed into a cell that is only visible through a microscope, so it's not visible with the naked eye, and you might wonder how that is possible. Well, the answer to that is in a chromosome, the DNA gets like coiled around some spools that are called histones, and it's almost like winding thread around a spool, so that the DNA can be made to take up less space, and you have the histone that has the DNA wrapped around it, and then there are other structures like proteins that help to support the organism. And when you get all of that together, that's called a chromosome. Okay, so the genetic material, the DNA wrapped around the histones with some other materials, give us the chromosomes. Now, the number of chromosomes varies depending on the organism. And it doesn't really, the number of chromosomes doesn't have anything to do with the complexity of the organism. So you can have an insect that has more chromosomes than a human being, even though the human being is a much more complex, more higher developed than an insect. So humans have 46 chromosomes, I think your book mentions that horses have 64. And then crayfish, which we'll dissect when we do the lab, they have about 200. They have 200 chromosomes. Okay, so again, the number of chromosomes doesn't depend on the complexity of the organisms, but each organism does have a different number of chromosomes. So remember that number. We're going to deal mainly with humans here in our discussion. So remember that humans have 46 chromosomes. Okay, so this is a picture. It'll come up. This is a picture of a chromosome. I'm going to get a bunch of them now okay, so this is a picture of a chromosome. So we see the chromosome here. What you'll notice, so this is an unduplicated chromosome, and the chromosomes have an area in their center that's called a centromere. And one of the things that happens in reproduction is that the chromosomes will duplicate themselves. So here's one chromosome, and that chromosome will duplicate itself. So we have two copies that are exactly like this one, and they will join together with each other by their centromere. So they'll join together there in the middle. And then we call the two different chromosomes sisters. So this would be one of the sisters, and I'll draw the other one with a different color. This would be the other sister, and they would be identical to each other. Just so this one gets duplicated, and you have two copies that are exactly alike. Okay, so that's typically what we think about a chromosome looking like. So let's begin looking now then at the process of mitosis. Okay, and let's go back to the whiteboard so I can write up a few things, and then we'll go back and look at some figures. So mitosis basically consists of 4 phases, and the first phase is called prophase. The second phase is called metaphase. Third phase is called anaphase. And the last phase is called telophase. Okay, so a long time ago, I learned that one way you can remember these different phases is that you, and I don't think this is too crude, so we'll go ahead and say it. It helps me to remember it. You pee on the mat. Okay, so pee on the mat. Okay, so prophase, metaphase, anaphase, telophase. So if that helps you to remember, so be it. If it doesn't, you can maybe think of a different way that would help you to remember the 4 phases of mitosis. In addition to these 4 phases, you also need to know that there is a phase that is called interphase. And Interphase is when the cell is in, this is interphase is where the cell spends most of its time. So in interphase, it's not duplicating. So you might think of it sort of as being in the resting phase. Doesn't mean the cell's not doing anything, but it's not being duplicated. So interphase is the cell just carrying on its normal functions. Okay, and then when the cell starts to duplicate it, it will go duplicate itself. It will go through these 4 phases in order. Okay, so let me tell you what happens in each of the 4 phases, and then we'll look at a picture to illustrate it. Okay, so the first phase is prophase. And what happens in prophase is that the DNA duplicates. And then there are little structures that I think we talked about when we learned about the parts of a cell that are called centrioles, and they also duplicate. Okay, so the DNA and the centrioles duplicate, and then these centrioles begin to form what are called spindles. Okay, so that's what happens in prophase. Okay, so let's erase the, no, let's not erase that, but let's go and look at a picture. Okay, so here is a picture of the steps of mitosis. So here is interphase. Color that we can see more easily. Okay, so here is interphase, and this is what the cell would look like when it is just carrying on its normal functions. It is not duplicating. I mentioned those centrioles. So here are the centrioles right here. Okay. So then what happens during interphase and during prophase, and prophase is shown here kind of in 3 steps, but what happens is the DNA duplicates itself, and so also do these centrioles duplicate themselves. Okay, so here we see the duplication of the DNA, and we also see the duplication of the centrioles, and then those centrioles start to form these connections between them that are called spindles. Okay, so this is a spindle, that's a spindle. Okay, so all these connections between the centrioles are spindles. Okay, so then, so we went from a chromosome that looked like this, and it duplicated itself. So now we have like the sister chromatids that we had looked at previously. Okay, and by the time you get to the end of prophase, those chromosomes are going to start lining up in the middle. Okay, they're not fully lined up in the middle, but they will start to line up in the middle. Okay, so prophase, the chromosomes and the centrioles duplicate themselves, and they form spindles that attach to the chromosomes. Okay, so that's prophase. Then okay, so that's prophase. Then if we go back to our whiteboard, then we would go into the next phase. So P on the mat. So the next phase is metaphase. And metaphase, meta means middle. Okay, so remember I said that by the end of prophase, those duplicated DNAs will start to line up in the middle. Well, in metaphase, they do line up in the middle. Okay, so you get the duplicated chromosomes that have lined up down the middle of the cell. I didn't draw that very well. It's not really down in the middle, but see if I can draw a better circle. So here's a circle, and those, let's make them red, one side red, one side red, one side red, and then to show that they were duplicated, we'll make the other side blue. Okay, so the chromosomes line up down the middle in metaphase, and then the other thing that happens is those spindles attach to the middle of the chromosomes. Make that black. So the spindles attach to the middle of those chromosomes, one on each side, and they start to pull on those chromosomes. Okay, so the spindles attach and they start to pull. Okay, so let's go back and look at a picture. Look at that picture that we had before. So now we are in metaphase, and you can see in metaphase that the chromosomes are lined up down the middle, and these spindles are attached to them, and they're starting to pull on them. Okay, so that's all that really happens in metaphase. Is because metaphase means middle, they line up in the middle. Okay, let's go back to our whiteboard. And now the next phase, so we'll erase this, phase number 3, P on the mat. So the next one, it starts with an A. So that's phase 3 is anaphase. And what happens in anaphase is that those chromosomes that were duplicated get pulled by the spindles so that they separate from each other. Okay, so if we had them lined up in the middle. And the spindles are attached to them, like so. Then what happens in anaphase is that these spindles pull on these so that the one half of the chromosome gets pulled one way and the other half of the chromosome gets pulled the other way. So what you end up with in anaphase then is that you get the reds over here and you get the blues or the other part of the chromosome on the other side, and they're still attached to the spindles. Okay, so the spindle, oops. Draw my circle. So these spindles will still be attached, but now they are the different halves of the chromosomes have been separated. Okay, so that's in anaphase, the two halves of the chromosome get separated from each other. Okay, so let's look at the picture of that. Okay, so now we look at anaphase here, and you can see that in anaphase, the sister chromatids get pulled away from each other, separated from each other. Okay, then the last phase is called telophase, and all that happens during telophase then is that the plasma membrane kind of constricts or pinches off so that these chromosomes that were pulled off and separated from each other then form two separate cells, and these are called daughter cells, and the daughter cells will look exactly like the parent cell that we started with. So we started with one cell here, and we now have two daughter cells that look exactly like it. So interphase is the resting stage, then it goes to prophase, and prophase, you duplicate the chromosomes, you duplicate the centrioles, and by the end of toward the end of prophase, those chromosomes line up in the middle with the spindles attached to it. Then in metaphase, they do line up in the middle, and you can see the spindles attached there. In anaphase, the spindles pull so that the two sister chromatids get separated from each other. In telophase, the plasma membrane pinches off so that you end up with two daughter cells that look exactly like they're exact copies of the mother cell that you started with. Okay, so that is mitosis. That is how we go from having one cell to having multiple cells that look exactly like the I'll go ahead and write up the last one here, which is telophase. And in telophase, the plasma membrane pinches off. And you end up with two cells. Okay, then each of those two cells will be in interphase, and each of those two cells will then go into mitosis, and each of them will produce two cells. So now from the initial one cell that we started with, we have 4 cells. Those 4 cells will go through mitosis, and then we had started with 1, and then we'd have 8, and et cetera, et cetera. That is how mitosis happens. Okay, let me introduce a few other terms, and those terms are diploid and haploid. And diploid is sometimes referred to as 2N, and haploid is referred to as N. So diploid refers to the many chromosomes come in pairs, and we just saw that. So chromosomes come in pairs, and those pairs are not exactly like each other when sexual reproduction is happening, but they are similar to each other. So when we say that humans have 46 chromosomes, the 46 is the diploid number. So the total number of chromosomes in a diploid cell is a diploid number. And the haploid number is that's a cell that only has 1/2 of each chromosome pair. Okay, so if you have a chromosome, and when we say that that humans have 46 chromosomes, they have 23 pairs of chromosomes. So chromosomes come in pairs. So the 46 chromosomes that humans have exist as 23 pairs. So so the diploid number would be the total number of chromosomes, and the haploid number is the number of pairs. So this is the number of chromosomes, and the haploid number is the number of pairs of chromosomes. So earlier we said that a horse has 64 chromosomes. So that would be, its diploid number would be 64, and then its haploid number would be 32. Okay, so the total number of chromosomes and the number of pairs of chromosomes. Okay, so if we think about a human having 46 chromosomes, and we said that in sexual reproduction, you have an egg and you have a sperm. Well, if this egg had 46 chromosomes, and if the sperm had 46 chromosomes, then when these join together, you would end up with 92 chromosomes. And that would not be compatible with life. That organism could not survive. So this will not happen. And so what has to happen before the egg and the sperm come together in fertilization is that you have to get the number of chromosomes in each of them down to half of their number. So this needs to get down to 23 chromosomes, and that needs to get down to 23 chromosomes so that the organism, the offspring that results will have 46 chromosomes. And this is what happens in the process of meiosis. Okay, we take, it's the making of cells that have half of the genetic information, so that when the male and the female egg and sperm come together, then you have a full complement of the number of chromosomes. Okay, so let's look then at the steps that are involved in meiosis. Okay, so this is meiosis, and a couple of things I'll point out right away, and that is that meiosis involves more steps than mitosis, those steps have the same names. They're called prophase, metaphase, anaphase, and telophase, but there is a meiosis 1 and there is a meiosis 2. Okay, so this top portion up here is meiosis 1, and then this bottom portion is meiosis 2. Okay, so let's look at the steps that are involved in meiosis 1 and meiosis 2. Okay, so just like before, interphase is kind of the resting phase. It's when the cells are not reproducing. And then in late prophase, what you get happening is that the chromosomes duplicate. They've duplicated and they are lining up in their pairs, and the spindle is beginning to form. Okay, then what happens in metaphase 1 is they do line up in the middle, just like we had seen before. They line up in the middle. And now here's a way that meiosis is different from mitosis. In anaphase, when the spindle starts to pull on the chromosomes, remember in mitosis that it pulled the different halves apart. Well, what happens in meiosis is it doesn't pull the pairs apart. It pulls the or doesn't pull the different. It doesn't pull the two halves apart. It pulls the pairs apart from each other. Okay, so here the red ones would be the same chromosome, so they get pulled apart from each other, and the green ones would be the same, and so they get pulled apart from each other. So we're not taking this and separating them into this and this. We're taking the pairs. So we have two green pairs, and we have two kind of reddish pairs, and that's what gets pulled apart. So the reds get separated and the greens get separated. That's what happens in anaphase of meiosis. Okay, then so they line up in metaphase, and then in anaphase they get separated, and then in telophase, same thing happens as before. The plasma membrane is going to kind of pinch off. So then at the end of meiosis 1, you end up with two cells, and each of those cells each contain half of the number of chromosomes. Okay, so this cell had 4 chromosomes, and these cells each now have only 2 chromosomes, but the chromosomes are still together as pairs. Okay, so this was a diploid cell, and now we have 2 haploid cells that have resulted. Okay, then those cells are going to go through meiosis II, and meiosis II now looks an awful lot like mitosis. So we start with these two. We start with 2 haploid cells, and in prophase, the centrioles duplicate and the spindle starts to form. In metaphase, they line up in the middle. Remember, meta means middle. So in metaphase, they line up in the middle. And then in anaphase, just like in mitosis, the pairs get separated. So we can see here the pairs are getting pulled apart from each other. So now what do we end up with? We end up with 4 cells. So in telophase, the plasma membrane starts to pinch off again, and we end up with 4 cells, each of which contains half of the genetic information. So you end up with 4 haploid cells. Okay, so we started with 1 diploid cell, and we ended up with, at the end of meiosis II, you end up with 4 haploid cells. So 4 cells that have no duplicate chromosomes. Each chromosome is different from each other. Okay, there's no duplicates like we see in the diploid cells. Okay, so that is meiosis, meiosis I and meiosis II. So that you end up at the end of meiosis I, you end up with 4 cells that are haploid cells. The chromosomes are not duplicated. So this is what happens in males and females, and that's what happens in the egg and the sperm. The egg, it happens in the ovary for a female, and it happens in the testes for a male, so that you end up with 4 cells that have half of the genetic information. So you end up with 4 haploid cells. Okay, so there are slight differences between males and females. So in a male, remember I said this happens in the testes. Well, in a male, those 4 haploid cells that result at the end of meiosis II. So here you had 2 haploid cells, but the DNA is still duplicated. And then when you go through meiosis II, you end up with 4 haploid cells in which there's no duplicate DNA. So these are called gametes, and in a male they would be called sperm. Let me go back to the whiteboard so I can write those words for you. So they're called gametes. In a male, those would be sperm. And in a female, so that this happens in a male in the testes. So meiosis takes place in the testes in a male, and in a female, the gametes that result are called eggs, and that happens in the ovary. Okay, so the one difference that I need to point out to you is that in a male, you end up with 4 cells, and those cells will then form a tail. It's like a flagellum in which they can move, and they can go in search of an egg. But all four of these cells are functional, and any of those cells could fertilize an egg. Okay, so that is in a male. Now in a female, when you go from meiosis 1, what happens during meiosis 1 is that when you end up with these two cells, one of the cells takes a lot more of the cytoplasm, and it also takes a lot more of the other organelles. So it's much bigger than the others, than the other one. And then when you go to meiosis 2, the same thing happens, where from this one you end up with a bigger one and a smaller one, and you end up with two smaller ones from this one. So this one breaks into a big and a small. So basically you end up with one large and three small. And in this is the only egg that is functional. These three are referred to as polar bodies, and they will disintegrate, and even if they get fertilized, they'll still just disintegrate, and they'll die and disintegrate. So unlike in a male where you end up with 4 functional sperm, in a female, when you go through meiosis, at the end of meiosis 2, you end up with only 1 functional egg cell, and you end up with 3 polar bodies, which will just disintegrate. So this one egg could be fertilized, but these 3 polar bodies cannot. Okay, so that's one difference between males and females. So let's go back to the whiteboard briefly. So then when we are done with meiosis, then the male sperm has 23 chromosomes. And the egg has 23 chromosomes. So when these come together, then you end up with the full complement of 46 chromosomes that make up a human being. Okay, so these are haploid cells. So a sperm is a haploid cell, an egg is a haploid cell, and then you end up with a diploid cell with the full complement of 46 chromosomes. I mentioned earlier that the sperm will develop a tail. So the sperm are mobile, they can move. Eggs cannot, so it's the sperm that has to go in search of the egg in order for fertilization to take place. Whether that is fertilization that takes place in the water or inside the organism, it is the sperm that needs to go in search of the egg, because the eggs are not fertile. Okay, one other thing that I want to talk about are viruses. So viruses, we've talked about bacteria, and that many bacteria are pathogens. Well, viruses are also pathogens. So viruses are responsible for the common cold. Viruses are responsible for the AIDS. So the HIV virus causes AIDS. And the reason we're talking about viruses is that viruses, they cannot duplicate themselves. So viral cells cannot duplicate themselves. They have DNA, some of them also have RNA, but they are unable to reproduce themselves. So technically, a virus would not be considered alive because they can't reproduce. So then how is it that viruses reproduce? So the way that viruses reproduce is that the DNA or the RNA from the virus will enter another cell, and it takes over the DNA of that cell, and it uses the DNA of that cell to duplicate itself. So even though they don't have the capability to reproduce themselves, they are able to take over the DNA of other cells, and then new viruses get made, and they will keep being made until there are so many of the viruses within that cell that cell ruptures. And then those viruses are then loose, and then they are able to go and infect other cells. So that's why, you can go from feeling fine one day to the next day having, a full-blown symptoms of a cold, because the cold virus will infect a cell, and then that cell will, the term is lyse, it will break apart and release those viruses so that they can then go infect other organisms. So colds, warts, chickenpox, the flu, mumps, measles, AIDS, all of those things are caused by viruses. And our bodies have defense mechanisms in order to fight or destroy infections, and those are called antibodies. So when your body has been exposed to a certain type of infection, the body will then produce these special proteins called antibodies that help to destroy the infection. And if a person gets a vaccine, what they are basically being given, in most cases now, it's an inactive form of the virus. Sometimes it's a weakened form, but most of the vaccines that are used today are inactive, so there's no way you can get the disease from them. So it's an inactive form of the virus that will then enter your cell, and then your body begins to produce antibodies against it. So that if you get exposed to that virus again in the future, then the antibodies are already there to begin to fight it. So you have the defense mechanism already in place even before you get exposed to the real virus. Okay, so that's a little bit about how viruses reproduce, because they can't reproduce in the usual way. They kind of hijack other cells' DNA and use those other cells to reproduce. Okay, I think we'll stop there. Let me see if I have any more pictures to show you before we are done. This figure just shows how viruses attach themselves. In this case, they actually attach themselves to a bacteria cell, and then they put their own DNA or RNA into that bacteria cell, and then when the bacteria cell reproduces, it's reproducing the virus. And then when there get to be so many of them, then the cell will rupture, and then more viruses get released so that they can then go attack other cells. Okay, so I think that is all we are going to do today. That is the end of the second part of Module 7 for biology.